So what I want to do today is, one, give you a picture of how I came to be an investigator, because it's perhaps a little circuitous, maybe not, and then move into a discussion of just one virus and the development of a vaccine for that virus that I've been involved in for actually since the middle 70s. And there's some failures and some interesting social aspects to it, but in the end, a successful vaccine, several successful vaccines have been developed. So to get started, I don't have any financial conflicts of interest. I wish I did with this vaccine, but I don't. But in fact, a number of the vaccines that I'll talk about, I invented or helped invent in a variety of ways, but I don't have any financial interest in them. So I was a history major in college. My mother said, "You're not going to get paid as a historian, get a trade." I figured I could become a doctor. And when I was in medical school, everyone said you need a good internship so you could have a good practice. And they said, if you did research, you could get a good internship. So I knocked on doors, and I worked in a laboratory that was studying what is now called hepatitis B virus, or serum hepatitis. And to make a long story short, nobody knew what caused that disease when I was working on it. And we developed a test for the disease. And to see whether our test was accurate, I would measure blood to see whether it was contagious. Nobody knew whether it was. And then my boss had this 1966 red convertible Mustang, and he would give me the keys, and I would drive around the Upper West Side and New Jersey and look for people who got transfused with this thing that was called hepatitis B. It was called Australia antigen at that time. And in fact, if you got a unit of blood that had Australia antigen in it, you got hepatitis with high frequency. And I'd ring the doorbell of somebody, and they'd open the door, and they were jaundiced. And that was, I would say, not good for the person who got hepatitis. It was good in the long run because we figured out how to test for it. But it was very exciting to prove that something caused disease. So that excited me a little bit. And then for those, I can see there are a lot of you who remember the Vietnam War. I almost never give a lecture to anybody who even knows that there was a Vietnam War. And again, if you're as a physician, I had a bad draft number, and I would have gone to Vietnam. The doctors were not only not deferred, once you graduated, you were bait. One of the ways of getting out of Vietnam was to go to the National Institute of Health and be in the Public Health Service, which is actually a branch of the Navy. Very competitive, as you can imagine, since no physician that I ever met wanted to go to Vietnam. Well, I went to the NIH and did more research, and I got more interested in it, and sort of the bug began to bite, and that helped me get a postdoctoral fellowship at Stanford. And because I had worked on a little hepatitis in medical school, I worked with some investigators at Stanford. They're both still around, both retired now. And we studied hepatitis B. And actually, I was able to be involved in the first use of interferon to treat hepatitis B. That led to a several billion-dollar industry. It's not really the drug of choice now. But that was very, very exciting, and by then I was hooked on research. It didn't hurt that my wife, who's sitting in the audience, wasn't that happy in Palo Alto during those years, in part because I didn't buy 2 cars and she had to take a train to San Francisco to work. So we moved back to the National Institute of Health, and I spent another 7 years there, and then came back to Stanford. But when I got back to the National Institute of Health, this is more or less the data on the incidence of killers of children under 5 in the world. And as you can see, they're all infectious diseases. And whoops, as you can see, diarrheal disease is actually the second most important killer of young children around the world, causing really almost 2 million deaths a year. And interesting, this data is from 2005, but that was also true in the late 1970s. And despite almost a whole century of investigation, the major cause of pediatric diarrheal disease around the world was unknown. People knew it must be a virus because they could take fecal filtrates from children who had diarrhea, filter them so that no bacteria were in it, give it to a volunteer, usually a medical student, and induce the disease. But they could not culture a virus. So it was one of the big medical mysteries of the world, because people knew there was a virus, but they couldn't culture it. And to give you a feeling of how much diarrhea there was in the world, this is a calculation that we made. There's less now, probably about half the amount of diarrhea, so it's maybe only 30 seconds that Victoria Falls will run with the diarrhea. But in any case, acute respiratory infections. I'm sorry, yes, acute respiratory infections. And in fact, as you know, for those deaths, that's mostly influenza, pneumococcal disease, and respiratory syncytial disease. So right around that time, in the early 70s, so there is basically from the 1930s to the 1970s, thousands of experiments done all over the world trying to grow viruses out of feces for the cause of this infantile diarrhea, all failures. And then simultaneously in Australia and in England and in the United States, it dawned on people, why not use an electron microscope and just look at feces under an electron microscope? What is kind of remarkable is that seems like the world's simplest experiments. People have been looking at things under a microscope for hundreds of years. Nobody ever thought to use an electron microscope in a diagnostic manner until that time. And this is what they saw. They saw one virus that I'm not going to talk about, norovirus. I worked on that a lot, but I'm not going to talk about it anymore, except for my next slide. And then they saw this beautiful virus, rotavirus, about 100 nanometers across. And actually, so this is just a little bit of diarrhea put on an electron microscopic grid and looked at. So you wonder where feces is. It looks like it's pure virus in the stool. And one of the things about this virus is it replicates to extremely high titers when children are infected. 10 to the 11th to 10 to the 12th infectious particles per milliliter of diarrhea. So one little bit of diarrhea could infect the country, basically. Very potent replicator. So in any case, all of a sudden, this agent that everybody knew exists was found. And the wonderful thing about rotavirus is that it was shed in such large amounts that it was real easy to detect it. And so that meant that epidemiology could happen very quickly. For a lot of viruses, it was very hard to detect, and PCR, which came along 25 years later, revolutionized detection. But for rotavirus, it was simple to detect. You could see it, you could detect it in many ways. But before I get to the rotavirus story, I just couldn't not talk to you about norovirus. You all probably know about norovirus because adults get norovirus, not just children. And it's the epidemic diarrhea. You get it from oysters. I don't know who here has eaten oysters and gotten a gastroenteritis. For those of you who remember George Bush having vomiting in the lap of the Japanese premier, that was a norovirus infection. You get it on cruise ships. When the cruise ship, everybody is sick, it's norovirus. It's a great virus to study. We don't have a vaccine for it yet, but I just don't want you to be confused with rotavirus. So another sort of interesting sidelight of the rotavirus story. So here's this virus that really causes a lot of disease in children. And when all of a sudden it was discovered, the veterinarian said, "You idiots, We knew about rotavirus for 20 years. It causes diarrhea in cattle, it causes diarrhea in pigs. There was already veterinary vaccines, and no human investigator of humans, and especially of pediatric diarrhea, ever thought to read the veterinary literature and say, gee, if there's something that's causing diarrheal disease in people, maybe there's an analog in the animal world. Well, it turns out that every mammalian species that has been looked at has rotavirus that infects that species and causes diarrheal disease in the young of that species. So it's ubiquitous everywhere, but the human one wasn't found until somebody looked specifically in human stool. And because it was shed in such large amounts and easy to detect, global burden of rotavirus was very quickly worked out. And as you can see here, this is older data, but this data still is after vaccination. My old slide, when I used to give this, rather than 215,000 annual deaths, were closer to 500,000, but it's gone down by 50% because of vaccination. But in any case, as you can see, the poor countries have many more deaths than the rich countries. And that's true for most infectious diseases. And it's not necessarily because the disease is particularly different in those countries. It's because access to care is different. And especially for rotavirus, because it's almost impossible in the United States to die from a rotavirus infection because you get hydrated. You die from dehydration. And by and large, almost every child in this country gets access to enough hydration so that they don't die. Very big cause of hospitalizations, but not death. And the reason there's so many, anybody have any idea why there's so many dots in India as opposed to other poor countries? Well, clean water. There's lots of places where you don't want to drink the water, India being one, but there's many other countries. Just more babies. So there's more babies that die. So India is the birth capital of the world. 24 million babies born a year in India versus about 3.5 million babies in the United States. So more babies that can die, more babies that die. So just to tell you clinical symptoms, any mom who has a baby and who wasn't getting vaccinated, the baby gets a lot of diarrhea, gets dehydrated, and there's a fair amount of vomiting, which adds to the dehydration. Rotavirus is different from the other diarrheal diseases of young children in that it's more severe. So if it's severe enough to go to the doctor or go to the hospital, it was almost always rotavirus in a country like the United States. But no other statistics. And it does cause a little diarrhea in adults. The data that I used to give before we had a vaccine is if you're in a family that has a child with rotavirus diarrhea, about 25% of the parents of a child with rotavirus diarrhea were going to get some sort of gastroenteritis from the child, but mild, a day or two. But now it's less because we're heavily vaccinated. So rotaviruses, I don't know how much virology I should get into here, not a lot, but the world of viruses can be divided up into RNA viruses and DNA viruses. Don't have any other form of genetic material. Rotavirus is an RNA virus. It can be divided up in viruses that have membranes around them and viruses that just are protein. Rotaviruses is one of those viruses that is just protein. And the RNA in the virus is kind of unusual. It's double-stranded rather than single-stranded, like DNA, and it's segmented. So rotavirus actually has pieces of RNA inside each particle, and each one of those pieces is a single gene. So it's got 11 pieces of RNA, and each one encodes a single protein. So these are very simple viruses. A virus like vaccinia or herpes viruses have hundreds of genes. Rotavirus just has 11. Influenza just has 8. Influenza can kill 40 million people in 1 year. Just 8 genes. Little virus, but capable of raising real havoc. in any case, the reason you should know whether a virus has segmented genome in pieces or not, when viruses have pieces of nucleic acid inside them, if two viruses infect the same cell, out of the progeny, just like when you have children, have a mixture of the genes of both parents. So that's one way viruses can make a very high level of genetic diversity very, very quickly. And that's true for segmented gene viruses. Influenza is the one you should know most about, but it's also true for rotavirus. And the rest, in my talk, I'll just briefly, the things to focus on. There's two proteins, Vp4 and Vp7. Vp4 here is this pokey thing that's sticking out from the virus. Vp7 is this yellow surface. Those are the two proteins actually that are targets of protective immunity. If you're going to make a vaccine, you want to make something that makes antibodies to those two proteins. And I won't talk about it much more, but this gene here, gene 5, makes a protein that is able to knock out your innate immune system. It's an anti-interferon gene. And I do a lot of research on that. I'm not going to talk about it today because it's not important for vaccines, not very important. So the other thing I want, speaking about vaccines, do you all know the story of Jenner and the smallpox vaccine? How he saw the milkmaids and they didn't get smallpox and they figured out that it was a cowpox. Well, it became clear very early that there were all these rotaviruses in animals, and an early observation, also made by me in this, is that animal rotaviruses from one species were very weak in another species. So when you have a baby, if you have a child at home, and you have a dog that has a dog rotavirus and the baby's crawling around on the ground, even if that baby gets some fecal material with the dog rotavirus in it, they don't get sick from the dog rotavirus. And the dog doesn't get sick from the baby's rotavirus. That's known as host range restriction. So we live in a globe, because these viruses are so shed in such large amount, there's a fecal veneer around the entire globe. It's thicker in India than it is in the United States, but it's in this room, it's everywhere you go. And believe it or not, it has human rotaviruses in it, it has dog rotaviruses, mouse rotaviruses, and if you had PCR, you could amplify them all up. But the only ones that are really going to make you sick are the human rotaviruses. And so the idea was maybe you could take an animal rotavirus, like the cowpox, and give it to people, and it would immunize them and be protective. And that was an early idea of a concept about a vaccine. One other thing I want to talk to you about, so immunity. So we're talking about immunity in people. A vaccine induces immunity to a disease. Well, this is sort of amazing. This is Thucydides, 400 years before Christ, 430 years. So he said, it was with those who had recovered from the disease, I don't know what disease he's writing about, that the sick and the dying found most compassion. This is sort of funny because you're compassionate if you're not going to get sick, it turns out. These knew what it was from experience, and they had now no fear themselves, for the same man was never attacked twice, or never at least fatally. That's immunity. So it's been known about for a very, very long time. And in fact, people who had smallpox never got it again. People who had polio could get it 3 times because there are 3 different polios. But basically, measles once and you're done. Hepatitis once and you're done for some types of hepatitis. So in any case, was that true for rotavirus? So this was an early study that simply said, if you're naturally infected in a baby, what's your chance of having a bad infection again? And in fact, what Thucydides said 2 millennia ago was true for rotavirus. 1 natural infection, about an 87% chance that you would never have a serious second infection. 2 infections, 100% immunity. So natural infection gives you immunity. Now, do any of you know a disease where natural infection doesn't give you much immunity? Influenza is one, right? Because the virus keeps changing all the time, so you get it your whole life. The common cold, rhinovirus, is another, because there's about 150 different types of rhinovirus, and immunity to one doesn't protect you against the other 149, so you keep getting it. But for lots, you can't, so those are really tough. Tough to make a vaccine, but it was obvious that you might be able to make a vaccine for rotavirus. And then the last bit of knowledge that we got before we started is we did, and I did, you're looking at a slide that represents about 15 to 20 years of my life, which is pathetic in some ways. But we did, I tried to say, what are those proteins that are on the virus that you want to make immunity to? And to make a long story short, those two proteins on the outside of the virus, antibodies directed at either of those would protect you from reinfection. So I couldn't figure out, still can't figure out which is better. So now we know that you can be immune. We know what the targets are. We know that animal rotaviruses are not likely to be cause a lot of disease in people. So what are we going to do? Well, what we initially did is take an animal virus, and I took several, but here I'm dealing with a monkey rotavirus, and I actually mated it with a human rotavirus. So I put those two human surface proteins, Vp4 and Vp7, the ones that you want to have immunity to, on top of the animal rotavirus, inside the animal rotavirus. And I said, maybe this virus will still be attenuated, weak in humans, but it has the two outside proteins of the human, so it'll immunize. We called that a modified Jennerian approach, so it was a little bit different than Jenner. And we commercialized it with Wyeth, and this is way back. This is in the 70s. We made a vaccine. Wyeth, a commercial, it used to be a big drug company. It's disappeared now. It basically bought. And the fact is, in multiple big studies, that vaccine worked, looked great, and it got licensed. So this was really wonderful. This happened to me right about the time I came to Stanford. A little bit, right after it got licensed in the early 90s, around the time I came to Stanford. So it was marketed and given to a little over 1 million babies in the first year here in the United States. Actually, then I did have a financial conflict of interest because I was getting royalties because I was part of the patentry of this vaccine. By the way, I was a government employee, so they cap the royalties that you can get as a government employee at that time. But it was enough, it was nice. But then here, this is a weekly that comes out from the CDC. All of a sudden, people noticed maybe some children that got vaccinated had what is known as an intussusception. Do you all know what an intussusception is? Do any of you know what an intussusception? There's at least one person. My wife knows what an intussusception is. Well, this is what an intussusception is. And it is the most common surgical emergency for children under the age of 2. Way more common for children of that age than appendicitis or inflammatory bowel disease. And what it is that the small intestine, you know what that is, and right at the end where the appendix is, sort of herniates into the large intestine, sort of like you could push your socks inside out. And when that happens, the blood vessels that are feeding this small intestine go in here and they get squished inside the large intestine. And when that happens, that bit of small intestine doesn't get blood, and if it doesn't undo itself, it dies. Bad news. Then you either need an operation or you die. And so this is a normal thing that happens to children. Nobody knows the reason for it. Actually, in the United States now, happens, 1 out of every 2,000 babies. And they used to operate to fix this. And now they learned, and this is kind of cute. I think it's cute. They just take the baby who has an intussusception, and the anus is over here. They put like a little air pump on the anus and pump air in, and they pop that open. And then there's no operation for the baby, and it works almost 100% of the time. It's a clever little idea. In other parts of the world, they don't do that. And the only way you can do that is if you have ultrasound and you can make the diagnosis by ultrasound. But in any case, so there were reports, because we have a system in this country that if there's if something bad happens after a vaccination, we try to gather that information. So this is the data that they got, and they found an increased incidence of intussusception in the first week after rotavirus vaccine. And you can see it right here. This is actually the data from the time. All of a sudden, you see these are intussusceptions that are occurring on, the regular intussusceptions before vaccination. Vaccination, all of a sudden, boom-o, a whole bunch of intussusceptions. And then, so this event, intussusception, seems to be temporally related to vaccination. That's a very good proof of cause and effect. And what the CDC did then, and I'll forever not forgive them for this. The CDC actually, when this happened, they discovered it, and they were under a fair amount of political pressure for other things. And so this was a great discovery for our CDC, and they made a big deal about how they were going to save the world from this dangerous vaccine. And they said relative risk is 25-fold. If you get the rotavirus vaccine, you have 25 times a higher chance of having intussusception than if you don't get it. And that was true for this 10 days after you got the vaccine. So that's relative risk. But is relative risk meaningful? So the opposite of relative risk is absolute risk. The real question is, if you get the vaccine, how much more likely are you to have an intussusception than if you don't get the vaccine in the next 2 years, let's say? There was no attempt to calculate that. So maybe these babies would have less than the background in the next 9 months. That is a possibility. Maybe the vaccine stopped intussusceptions that were going to occur or caused them to occur where they were going to occur later. And so my mother, who is now died at 96, always had her wits about her, did not get this concept of relative risk and absolute risk. And so I think I explained it to her by describing, if you're standing on the sidewalk and the traffic, and there's a street in front of you, and even if there's a red light, when you cross, your risk of getting bumped off by a car when you cross the street, your relative risk is way higher than if you stayed on that sidewalk your whole life. But your absolute risk is de minimis no matter what. So there are a few unusual people who will never cross the street because they're worried about that increase in relative risk. But basically, the relative risk is meaningless in that case because it is what's the real risk. And in this case, as you'll see, relative risk was the only thing that was used to evaluate this vaccine. So after all my brilliant colleagues at the CDC conferred, there was a whole national meeting, and the vaccine was withdrawn, taken off the marketplace out of the marketplace very quickly. Wyeth, at the same time, the manufacturer of this vaccine, had made the pill fen-phen. Do any of you remember fen-phen? It was a diet pill, and people thought it might injure the heart. And they were involved in estrogen for menopause. So Wyeth was being sued right and left for things, and they said, we're getting out of this vaccine business fast. And it was withdrawn. And the interesting thing was, as soon as the United States withdrew it, the rest of the world, and especially India, South America, Sub-Saharan Africa, all of them said, we don't want this vaccine. If it's not good enough for the United States, it can't be good enough for us. even if the risk of rotavirus is much higher in our countries. And basically, that was also a political decision that people didn't care. You couldn't politically give a vaccine in a less developed country, even if it would do a lot of good, if the United States said it's not good enough for us. So that was bad in my mind. So this is just a risk-benefit that I and others did at the time in the United States. So you just see here, no doctor visits, hospitalizations, intussusceptions, and deaths. And in the United States, since there were so few deaths from rotavirus anyway, it's a wash. Basically, maybe 3 more deaths in the United States if you take away rotavirus vaccine than not. Hard to say. But for hospitalizations and doctor's visits, huge differences. But this type of calculation and discussion was never made. But this is another calculation we made for the less developed world, and that was how many deaths were there because a vaccine wasn't available for the 6 more years until a new vaccine could come along. Probably more or less, let's say somewhere between 250,000 and 500,000 babies died because of the decision that what's not good enough for the United States isn't good enough for India, even if it kills a bunch of babies. So unusual politics. Fortunately, at the time that vaccine was released, several other large pharma companies, Merck and GSK, were in the process of making their vaccine, and they had 2. And when am I supposed to finish this? About what time? I'm not hearing. No, I know. I mean, it's about 6:15 to 6:15. Okay, we're fine. So GSK and Merck were making their vaccines, and I won't get into. They're very different ideas about a vaccine. The Merck vaccine is very much like that rotavirus vaccine from Wyeth. In this case, they used a bovine donor rather than a monkey donor, but it's a virus, a modified Jennerian vaccine. And they were still worried, as were we, that those proteins were different from one virus to another. So they made 5 different vaccines with different surface proteins so that they could stimulate immunity to all the different types of human rotavirus. And GSK made it the really old-fashioned way, the same way Albert Sabin made the polio vaccine, same way the German measles vaccine was made. They just grew the virus in cell culture over and over again, and when you do that, the virus mutates to grow better in cell culture, less well in people, and eventually it becomes attenuated, and that's what happened. So, and sure enough, both vaccines were effective and highly effective in the United States and the Americas, and even in South America, which is really, South America is closer to a developed country in its burden of disease than sub-Saharan Africa and Asia. So they worked, and this is to show you how they worked. So here you have the impact on all-cause and rotavirus-specific gastroenteritis. So, by the way, so this is also a nice way to give you 2 terminologies when you evaluate people who talk about new medicines, and that is efficacy versus effectiveness. So here is efficacy. This is rotavirus, and then the vaccine comes in, and you see less rotavirus cause gastroenteritis. So the vaccine is effective against rotavirus diarrhea. But if you're a mom, what do you care whether your baby has rotavirus or not? All you care about is, does my baby have less diarrhea? And so that would be effectiveness rather than efficacy. And so that's all-cause gastroenteritis. And you can see that in the United States, if you didn't look for rotavirus, you just said, is gastroenteritis or diarrhea going down? the rotavirus vaccine effectively treats that. That's effectiveness, and that's a much better measure of impact of a medicine. So I would simply say to you, as you read about new cures and new medicines, they always show you efficacy. They say, here's the rarest of the rare cancer, and in this cancer, this drug is 80% effective. And if you said, what's the effect of all cancers? No effect. You couldn't possibly see anything. And of course, it's useful. If you happen to have that rare cancer, that drug is useful and meaningful. But on a social level or a societal or a public health level, it's not the same thing. And you should think in terms of both. And the real effectiveness is in mortality. Because some people will say, what's the big deal? A baby has the poops. That's not a big deal. But in many parts of the world, the baby dies. And here, luckily, they were able, in a place like Mexico, to actually measure infantile mortality, and that goes down with the rotavirus vaccine. Really nice proof that this vaccine is effective in affecting that's a very hard endpoint, mortality. It's not easy to measure sometimes. So around 2011, the conclusion was we had two very effective rotavirus vaccines. They were also safe. So what does the FDA say about an intervention? What you have to show to the FDA is it's safe and effective, both. And here we've proven that. By the way, in China, until very recently, you did not have to show effective, just safe. So it's really sort of interesting as a proof. You could market your product with no proof of efficacy. And for both vaccines, because of the problem of the original rhesus rotavirus vaccine, that it caused intussusception, remember that very high relative risk, they made Merck and GSK do huge studies post-licensure to show that there was no increase in intussusception. Pre-licensure, they made both companies do 100,000 baby safety study to see that there was no increase, and that's unheard of for any drug, to say you have to study 100,000 people to prove it's safe. And they looked great. So they said, so nobody, and I, who was responsible for this intussusceptigenic, in part, virus, could never figure out why this one caused intussusception and the other ones didn't. It didn't make sense to me. But a couple of other things. When you vaccinate, you really have to look at what the effectiveness is in many ways. So these are just two unexpected findings after vaccination. The first one was that, in fact, there is some rotavirus disease in older children and adults. As I told you, those people weren't getting vaccinated, but their rate of rotavirus disease fell because the children were being vaccinated. So this was a herd immunity effect. You get rid of the disease in the people who are causing it, and the whole herd is protected. And there's some thought that that's true for influenza. It's true, and it's heavily true for pneumococcal disease, where vaccination, that's a bacteria, vaccination of young children made a big difference in elderly for pneumococcal disease. So that's a wonderful side effect. And here's an unexpected side effect, that actually seizures went down in association with rotavirus. And nobody knows exactly why, but the incidence of seizures went down. One, rotavirus causes fever, so maybe a decrease in the total number of fevers in young children would lead to a decrease in the number of febrile seizures. But also, sometimes rotavirus might cause actually infection in the CNS. So it's not known, but seizures just went down in children. So it's in part a seizure medicine. So how well will this vaccine work in less developed countries? So I already showed you that the burden of disease, it's all well and good, and the companies make money by selling the vaccine in the United States. By the way, just so you know, Merck really controls the US market now. That's what they wanted to do. They had their eyes on the ball, and the ball had a $sign in it. And most of the rotavirus vaccine sold in this country is Merck. GSK had a more global point of view. Most of the rotavirus vaccine that is sold by Big Pharma in South America and in Asia is GSK, and they're still duking it out. So here are some moderate and variable efficacy of rotavirus vaccines in Africa and Asia. These are trials that came out about 10 years ago, 8 years ago, and both the Merck vaccine and the GSK vaccine. And you can see, before, remember, we showed efficacy was somewhere between 80% and 90%. Here the efficacy, 64% ranging from 40 to 80, 51% ranging from 13 to 73, 62% so by and large in the less developed countries, I would say in poor Africa and poor Asia, rotavirus vaccines, the GSK vaccine and the Merck vaccine, I think they settle for 50% efficacious in preventing severe diarrhea versus in Finland, the cleanest country in the world, where the rotavirus vaccine is over 90% efficacious. However, if you say, how many deaths do you prevent? You prevent infinitely more deaths at 50% efficacy in India than with 95% efficacy in Finland. So if the idea is to prevent death, you settle for the lower percentage but the higher population. So there it is, and we don't know why that data is, but that's been reproducible in, I would say, over 70 studies around the world. That the richer the country, the better the vaccine works, the poorer the country, the less well it works. And here is a summary slide. We can talk about later why that might be the case. I can tell you that there's been tons of grant money to solve this riddle, and I am 100% positive that none of it has paid for a good answer. But if you need a grant, places like the Gates will give you lots of money to tell them the answer, and God only knows what it is. So what about intussusception? So I've told you already that those vaccines were licensed as safe and effective, and they passed this 100,000 test. But now, when you're giving Rotarix or Rotatec, the two commercial vaccines, to whole countries, you can do gigantic tests of safety. You can get the data on the whole United States or all of Australia. And when they did that, what do you know? Both of these vaccines cause intussusception. Now, what's the difference? Well, there's two differences. One, when this data has come out, you've already proven the effectiveness and efficacy of the vaccine all over the world. So now you're saying it's causing some intussusceptions, but you have tremendous data on how useful they are. When it happened back in the late 90s, there was not a lot of data on how useful it was, so it was easier to torpedo them. Two, people would say, well, these don't cause intussusception as much as that first vaccine. And I'm not sure that's true or not true, because they've never been compared head to head. Here, the rate of intussusception, the excess rate of intussusception in children who get these vaccines is more or less 1 out of every 50,000 to 1 out of every 100,000 children gets intussusception. Do any of you know well, some of you have grandchildren, they all get a polio vaccine. They get the Salk vaccine. They get a shot. They don't get the Sabin vaccine. I got the Sabin vaccine. I bet a lot of you did too. That was the one that came out first. Why don't we give the Sabin vaccine in this country anymore? Anyone know? There's somebody who knows.
It's because in 1 in a million cases the child gets polio.
Right. So Rotavirus causes intussusception in 1 out of every 100,000 children who gets the vaccine. The polio vaccine causes paralytic polio in about 1 out of every 1,000,000 children that gets it. And in a country like the United States, with a birth cohort of 3.4 million, that means every year, if you were given the Sabin vaccine, 3 to 4 children would get paralytic polio, and everybody else is protected. That amount of polio was unacceptable publicly. In a country like India, with a huge greater burden of disease from paralytic polio, and also not as much money to pay for vaccine. The Sabin vaccine is way cheaper to make than the Salk vaccine. The Sabin vaccine is still being used, but as we begin to get rid of polio completely, we're going to have to get rid of the Sabin vaccine because occasionally it becomes virulent. So in any case, both vaccines caused intussusception, but here's a risk benefit, and that data was convincing. And nowhere in the world did anybody say, we need to get rid of this vaccine because it causes intussusception, because it did so much good. So I would have had my royalties. I would be a wealthy man if it had happened at the right time, but the fact is the vaccines worked. So our last issue that I'm going to talk about very quickly is a final issue for vaccines all over, and that is cost. So it's interesting. Vaccination is one of these things that it really, generally, you give it to everybody. So for example, the flu vaccine. I hope you all get your annual flu vaccine, even though people badmouth it all the time. It does protect you varying degrees every year. But that vaccine is sold as a commodity. It really, I think to make it is about, to get it is less than $10 a dose. So there's no big profit in making a flu vaccine. For the pneumococcal vaccine that Pfizer makes, it's a couple of $100 when you get that there. And if everybody gets it, it's a several billion-dollar venture. But Merck and GSK, up until very recently, were selling the rotavirus vaccine for like $100 for every baby, and places like the United States were forking it out because it was good. But India can't afford that. And basically, to be honest with you, those companies weren't really interested in producing a dirt cheap, safe and effective vaccine for less developed countries. And as much as this is an argument I would have with the Gates Foundation, basically until we enable the ability to make inexpensive, safe vaccines in the less developed world, for the less developed world will continually get ripped off by the developed world for the cost. And that differential is made up by people like the Gates Foundation that subsidize vaccination in the third world. I don't know about you, but I'd much rather have India make its vaccine itself and sell it to itself than have Gates say, I'm a great man, and I'll give you an extra $7 so you can buy your vaccine, because that will end at some point. In any case, that's my own personal point of view, but fortunately, we figured maybe we could start a company in India, no financial relationship with the company, but help a company make a vaccine in India for India that was cost effective. And so we did that, and I won't go into it. It took about 15 years, starting from ground zero with no factory, no nothing, to a big company now. And in fact, there's the paper, 2014, showing that the vaccine that we made in India was safe and effective and had about just as you would expect, about a 55% efficacy. It turned out, this is sort of an unusual story and a sidelight a little bit. In a variety of neonatal nurseries around the world, they get colonized with a rotavirus And in each neonatal nursery, that virus is different than everything that is circulating around. And the children in the neonatal nurseries are almost never sick from that virus, and they look like they're protected later in life. So this vaccine is made from one of those viruses. And it turns out that it is sort of a Jennerian vaccine. That is, one of the proteins, actually one of the proteins on the surface, comes from a bovine rotavirus. So somewhere along the line, a little Indian child got infected with got some bovine feces and some human feces in them, and a new virus came out that was mostly human but partially bovine, and that virus is attenuated in children and immunizes. And look, we beat out Merck and GSK, almost threefold of the cost, and so this vaccine now is being given, last slide, across India. There's Modi. Government of India will provide a rotavirus vaccine to all Indian children. It's being rolled out, I don't know currently, but India has an awful lot of states, but it's at least half the states now have this in their childhood immunization program. And it's just got WHO pre-approval, which means Gavi and WHO can buy it to sell it in Africa also at that cheap rate. So that's the rotavirus story. Thank you.
Well, thank you, Harry. Thank you so much for this insightful talk. And now we have time to take your questions. And I'll be happy to walk around and take the questions with the microphone. So who wants to go first? Yeah, okay.
She's a plant.
So you said that it was very difficult to grow the virus in laboratory, but then the GSK, they did that, right?
No, so a lot of what I said, I glossed over many things. So all human rotaviruses are very difficult to grow in a laboratory until you learn a trick. And the trick is that you need to add trypsin. Trypsin is a digestive enzyme made by the pancreas. And actually, rotavirus, that surface protein, Vp4, needs to be cut by trypsin to allow it to grow in cell culture. And so once you learn, and this is actually a trick that other viruses need. If you add trypsin, you've solved it and you can grow human rotaviruses.
Oh, I'm sorry. Thank you for your interesting lecture. I never understood how a vaccine against a virus can stay effective over the long term with the viruses constantly changing. How does it keep up with the changes?
Well, so that's a very good question, and the answer is that some viruses are constantly changing. Some viruses are changed at one time. That is, there's rhinoviruses, the common cold virus. They're all rhinoviruses. If you looked at them under the electron microscope, they'd all look identical. They have sequence differences in their surface proteins. So each one, if you met them on the street and you were a virus, you'd say, you don't look exactly like me. And the immune system sees one and doesn't see the other. So when the immune system makes an immunity, so when you get your first common cold, you're pretty immune to that forever. But as I said, there's 150 other ones that are ready to give you a cold. Influenza, when you get it, actually there's good studies of this. The first influenza infection that you ever got, if we had some of it and gave it to you now, you're still immune to it. But you're not immune to the mutations in influenza that occur every year that make you susceptible. But not every virus does that. Measles never changed, has never changed in the history of what we know of measles. So if you're 100 years old, well, no. The first child who was vaccinated for measles, if they're still alive today and they got a bunch of measles in their nose, they'd be immune to it. The first person who was infected with measles. So every once in a while there's an epidemic of measles because somebody comes into the country and we don't vaccinate as well. But I had measles as a kid. I could bathe in measles and I would not get it. So it has to do with the same thing with hepatitis B. If you happen to have gotten hepatitis B and survived, you're never going to get infected with it. So now the next question you asked is, how is that possible that your immune system remembers what that virus looked like 70 years later? It's a hard question. We still don't know exactly why that is happening. And it doesn't happen with every virus, but vaccinia. If you got vaccinated as a child in 1947, or that's when I probably got vaccinated around, I still have immunity to that immunization. And I won't tell you how long ago that was.
Toward the end, you mentioned the neonatal mutation that was produced by the child being exposed to the bovine virus and blending it with a human one, producing something less vicious. Has there ever been an effort to crowd out a virus by introducing a less evil competitor and just...
I don't know of such. So when I first started talking, again, what I wanted to say is, you have swallowed in your life, I hate to tell you, but a lot of animal feces, and in that there were rotaviruses, and you were not infected by them most of the time. It depends, of course, a little bit on dose, but most people in this country don't get a gigantic dose. In India, you might get a bigger dose of animal, but there are huge, one of the first slides I showed, basically you need 10,000 times more monkey virus to infect a mouse than a mouse virus. So that's a huge difference in dose. So it's not a good strategy. In cell culture, they're on equal terms. And the reason you need much more of the non-mouse rotavirus than the mouse rotavirus is that the mouse rotavirus knows how to evade the innate immune system, the part of your immune system that makes interferon and other things. And the monkey rotavirus doesn't know how to shut down the mouse's innate immune system. I didn't get into that, so you didn't miss it.
Hi, thank you so much for your talk. I'm over here. Since it's so expensive to have a vaccine in India or in like developing countries and the efficacy is only slightly over 50%, would it be more cost effective and maybe like decrease the number of deaths if we focus on like implementing intervention programs such as giving them oral rehydration solutions? Would that actually decrease more deaths than say promoting vaccines?
So this is a great question. There's all sorts of people who are interested in almost everybody loves babies. And almost everybody doesn't want to see babies dying. So you're always a good guy or gal if you're working on that. And there have been competing forces of, we don't need vaccines, let's get clean water. Let's just promote breastfeeding. That's another one of those. And it turns out for rotavirus, okay, I'm just talking about rotavirus. Remember I started at the beginning saying shed at 10 to the 10th to 10 to the 12th particles per gram of stool? Well, there's no amount of sanitation. You could clean your house continuously and there's still rotavirus there. I don't know how many of you really have any understanding of how many things 10 to the 12th per gram is, but you can't wash it away. It's impossible. So the clean water will not, and in fact, the beautiful proof of that is babies in the United States with great water, flush toilets, get infected just as frequently and just as early as babies in India living, because even though there's maybe 1 million times less rotavirus, there's still tons more than you need. So that's the for rotavirus. For other things, it might be the case. Like for cholera, you're correct. And breastfeeding is an interesting one. Of course, I want to be politically correct here. So I am not saying anything bad about the value of breastfeeding. But there have been a lot of studies, again, for rotavirus, that have not been able to show that breastfeeding has with or without breastfeeding, has a demonstrable effect in lowering the burden of disease of rotavirus. Also, probably because the environment just overwhelms it.
Hi. Do you know how or why rotavirus vaccines cause intussusceptions?
That's a great question. I wish I did. So why don't I know? My little invention got knocked out of the box because of intussusception. I'm a gastroenterologist. I should be interested in it. So I'll tell you what the problem is. At the most, the vaccine that caused the worst intussusception causes it in 1 out of every 10,000 children. Very hard to study something in 1 in 10,000, especially when it's been taken off the market so you can't give it to any children. And the other ones, the current ones, are 1 in 100,000. So that's almost hopeless. So what do we usually do? We have an animal model. We get a model of it. Nobody has been able to produce an animal model of every animal gets rotavirus disease, can get it. No one of them has shown that it, but again, it may be such a low frequency event that it's almost impossible to model in a laboratory.